Hello everyone. Today we are going to talk about the Rapaport-Leubering cycle. So the Rapaport-Leubering cycle is the supplementary pathway to the glycolysis, which is operative in the erythrocytes. So in the erythrocyte, basically this cycle operates. So this cycle mainly concerned with the synthesis of 2,3 bisphosphoglycerate. This is the key substrate. This is the key substrate. This is the key uh, synthesis of the Rapaport-Leubering cycle. So 1,3 bisphosphoglycerate is produced in the glycolysis is converted to 2,3 bisphosphoglycerate by the enzyme 2,3 bisphosphoglycerate mutase. Again, I am going to tell you. If you don't know the glycolysis pathway, I want you to read that glycolysis pathway, and I want you to note down that glycolysis pathway before reading this Rapaport-Leubering cycle. So, talking about the Rapaport-Leubering cycle, after you have known all the steps of the glycolysis, um, then only proceed to this cycle. From the glucose, the glyceraldehyde 3 phosphate is produced, and from the glyceraldehyde 3 phosphate, by the action of glyceraldehyde 3 phosphate dehydrogenase, the 1,3 bisphosphoglycerate is produced. So on the 1,3 bisphosphoglycerate, upon action by the 2,3 bisphosphoglycerate mutase, 2,3 bisphosphoglycerate is produced. Again, this 2,3 bisphosphoglycerate can be converted to 3-phosphoglycerate by the action of the enzyme 2,3 bisphosphoglycerate phosphatase. You have to know this very well. So I am showing here the Rapaport-Leubering cycle for synthesis of 2,3 bisphosphoglycerate. Now let us discuss the significance of 2,3 bisphosphoglycerate. More detail on the hemoglobin and oxygen, I'll discuss that in the respiratory physiology in the oxygen dissociation curve. But here I'll just describe briefly about the significance of 2,3 bisphosphoglycerate. First of all, let us discuss about first significance of the bisphosphoglycerate. The production of the 2,3 bisphosphoglycerate allowed the glycolysis to proceed without synthesis of the ATP. This cycle is therefore the shunt pathway of the glycolysis to dissipate or waste the energy that is not needed by the erythrocyte. That means excess energy waste is dissipated by this cycle. But you have to know that the 2,3 bisphosphoglycerate is not a waste molecule in the RBC. The 2,3 bisphosphoglycerate combines with the hemoglobin and it decreases the affinity of oxygen to the hemoglobin. That's why in the presence of 2,3 bisphosphoglycerate, the hemoglobin unloads more oxygen to the tissue. There is more unloading of the oxygen from the hemoglobin to the tissue. So the increased amount of 2,3 bisphosphoglycerate in the RBC is seen in the various hypoxic condition, because in the hypoxic condition you have to know that more oxygen is needed in the tissue. So due to decreased affinity of oxygen to the hemoglobin and in the presence of 2,3 bisphosphoglycerate, the hemoglobin unloads more oxygen to the tissue. That's why in the various hypoxic condition, like in the high altitude, in the fetal tissues, and other various hypoxic condition, the 2,3 bisphosphoglycerate is seen more. And another is the glycolysis in the RBC is linked with the 2,3 bisphosphoglycerate production and oxygen transport. So in the deficiency of the enzyme hexokinase, the glucose is not phosphorylated. Hence the synthesis and concentration of 2,3 bisphosphoglycerate are low in the RBC. The hemoglobin exhibits high oxygen affinity in hexokinase defective patient. On the other hand, in the patient with pyruvate kinase deficiency, the level of 2,3 bisphosphoglycerate in the erythrocyte is high, resulting in the lower oxygen affinity. So this is the main thing you have to know in the Rapaport-Leubering cycle. You have to know the pathway of the Rapaport-Leubering cycle, and you have to know the significance of the 2,3 bisphosphoglycerate. The main significance of 2,3 bisphosphoglycerate is, you have to know that it is the shunt pathway in the RBC, and the 2,3 bisphosphoglycerate act on the hemoglobin to decrease the affinity of oxygen to the hemoglobin, so that the hemoglobin unloads more oxygen to the tissue in the hypoxic condition, in the various hypoxic condition. So this much for today. Thank you.